All right, let's talk about muscle contractions. So first of all, we're going to start off with the sliding filament theory. This is the theory in which thin filaments slide over the thick ones. This is going to pull those Z discs, or the ends of the functional sarcomere, closer together. When all the sarcomeres in a fiber do this, that entire fiber is going to get shorter. This in turn is going to pull on the endomycin, then the perimycin, and finally the epimycin, and then the attached tendon. And this in turn is going to pull on the bone, and we will have movement. Now, one fiber is not going to be enough, but we'll talk about that in a bit. So, one of the things that we do need to keep in mind when we have a cell muscle fiber, it is composed of many sarcomeres. These sarcomeres are going to contract as one. So it's not going to be one individual sarcomere, it's going to be all of them. In turn, that means this fiber will contract as one unit. The number of fibers that are actually contracting will determine the force of the contraction in the entire muscle. If we have enough force, we can move whatever's at the end of that tendon, be it your forearm, a finger, a leg, whatever the case may be. We actually divide this contraction process into 4 steps: excitation, contraction, coupling, contraction, and then finally relaxing. There is something that is known as the ratchet effect. This is also referred to as the actual contraction process. Also referred to as cross-cycle bridging, depending on your textbook. And basically what this is the action potential, the excitation portion of this. The action potential has been generated, it has traveled down that T-tubule, and it has triggered the release of calcium into the sarcoplasm. This calcium in turn is going to bind to the troponin, which is on the tropomyosin. When calcium binds to troponin, it's going to move the tropomyosin away from those myosin binding sites, because remember, up till this point, those have been hidden. When those myosin binding sites are exposed, myosin is going to attach. It's then going to cock, that's also known as power stroke. It's going to release, and this process is going to repeat. These will repeat over and over and over again until calcium has been removed from the sarcoplasm and or the ATP, remember that's our energy supply, is exhausted. So these two things need to occur, calcium plus energy, in order for this cross bridge to occur. And this cross bridging will occur all the way down the length of the thin filament. Now, if there's still calcium, if there's still energy, it will maintain that bridging at that final spot, keeping that contraction. All right, here is exactly what I just mentioned. Myosin binds to actin. It cocks, otherwise known as the power or working stroke, detaches. We bind new energy, and then we prime ourselves back up. I may need to remove this slide. I don't know if this animation still works. But if it does, you will see this when you're pulling this up on your computer. Here's that troponin-tropomyosin complex. Here it is when calcium is binding, and then the shift. We do have some other animations for you to watch. When I checked these at the beginning of the semester, these sites were still active. So that may have changed though since But here you can watch that in motion with other explanations, hopefully one of which, if mine didn't, resonates with you.